Hi, everyone, and welcome. This is Caroline with Fiveable, and tonight we're going to talk about nutrient cycles in succession. I can see already that we have some stress and anxiety about the AP exam, and I totally understand. At the same time, though, as much as you can, try not to stress too much yet. The good news is that we've kind of worked through the progression of most of our content, which is awesome. I always try to, in my class and with this podcast, I really like to front load and kind of work quickly through material the first time, knowing that you're not going to remember everything, because how could you? And you're probably in other AP classes, and you're definitely in other classes, right? So this is not the only thing on your plate. But on the same vein, we'll have a lot of time and multiple weeks of review. So like next week, we'll review all of ecology, and I'll start to tie in some of the AP standards and big ideas, and we'll talk about some strategies with free responses and multiple choice. And I have lots of tricks up my sleeve that range from, just doing the short free response before the long free response to the ordering of the multiple choice questions. So try not to stress too much. I will, and obviously your teachers will too. Sorry, just falling all over the place. But I'll try my very best to make sure that you feel comfortable and prepared. And also, obviously tune in, because during review, things are very boring and not helpful if you're not asking questions. So do tune in and come with questions prepared, even if it's not what we're talking about that night. We can certainly talk about it in more detail. And if you ask, a lot of questions about something one week, I can prepare to review some of those things the week after. So just utilize the resources best you can, and know that we'll prepare you. And again, I've intentionally left a lot of time and a lot of weeks for review, because your brain isn't going to remember everything. And that's, what review is all about, and helpful tips and tricks to come. Okay, so try not to stress too much. I know that's easier said than done. I have certainly been in your shoes. Okay, so we're going to get started. And again, we may, this may not take the whole hour tonight, and therefore if you do start to have questions, I can start answering things. I mean, obviously it's easier if I have those questions in advance, just so that I can prepare pictures and slides with information on them, as opposed to just talking at you. But I can certainly talk at you with anything that you guys want to, if we have some extra time tonight as well. Okay, whenever my screen decides to load, which could be forever. Here we go. We'll go ahead and get started. So I know when you think of exciting things to talk about, probably the first thing that comes to your mind is nutrient cycles. You're like, yes, let me talk about the water cycle. I can't wait. I'm jazzed, right? Probably not. But nutrient cycles are really important, and honestly, more important than the cycle itself is the rationale behind why we need them. So we're going to talk a lot tonight about macromolecules, and it's been a minute. The last time we talked about macromolecules, it was like the beginning of the school year probably, and a lot of you probably hadn't tuned into sessions yet. So you can rewatch those sessions if you get a Fiveable Plus membership. But we'll certainly review some key factors with them today, because that's, again, some of the most important part about nutrient cycles. Okay, so again, nutrient cycles. So all matter on Earth cycles through various states. I'm sure you've heard this before, right? But both matter and energy cannot be created nor destroyed. They're going to move differently through an ecosystem, and I'll talk about that in a second. But energy, matter, whatever we have on Earth has always been here. Right? So everything that we are made up of, everything on Earth is made up of, it's always been here. We're just changing the form of it. We're combining it. We're creating new things, right? But the atoms that exist on Earth always have. They just get rearranged into fun new things. And again, the same goes with energy. But as far as ecosystems function, so last week we talked a lot about energy when we talked about trophic levels, and how energy flows through an ecosystem with food webs, etc. So energy flows, as I just said, through an ecosystem, meaning it's mainly moving in one direction. So you can see energy here is really just traveling from the sun to the producers, through our various levels of consumers, through, decomposers and all that stuff, and then out the other end. And the out the other end is energy being converted into heat, right? So we did talk about this last week with each level from producer to primary to secondary to tertiary consumer. We only gain 10% of the energy from level to level, and the rest is lost, not actually lost, because again, energy cannot be created or destroyed, but transformed into heat. And so that flow actually just, again, moves in one direction, meaning the sun is not getting any energy from plants. Plants are not getting any energy from herbivores, etc., which makes sense, right? But it's an important fact to state. On the flip side, nutrients are much more easily to visualize because they do cycle through ecosystems. And so we can actually more tangibly view carbon or water or phosphorus or nitrogen moving and transforming into different forms throughout the ecosystem. And those processes are really important because of what they give the consumers and the producers in the process. And again, that's why we're going to talk a lot about macromolecules today, because that's the most important aspect of the nutrient cycle. Okay, so what are our most important nutrients? So we're going to go through a handful of them. So hydrogen and oxygen, we love them. Major source, easy, water, right? H2O. And then organic molecules. So it's really important to know, you absolutely need to know the atoms that make up our 4 major macromolecules: carbohydrates, lipids, nucleic acids, and proteins. So oxygen and hydrogen are in all 4, right? Oxygen, hydrogen, and carbon are going to be in all 4. So that's majorly important as well in the development of all of our macromolecules, which obviously make us up as humans. Major uses of hydrogen and oxygen, so it's obviously involved in organic molecules when we're making new ones. And water is also a major component in photosynthesis. So water helps us do many, many things, but we're going to talk mainly about plants here as our primary producers, which are giving us all of these organic molecules. So water is going to be a major component in photosynthesis. So I have a challenge question for you. Does anyone know, and you can just type into the chat box, does anyone know exactly how water is used in photosynthesis? Does anyone know the actual process? Give you like 30 seconds to think. If anyone knows the answer, you can just type it into the box. Yeah, slogan, you win. Electrons for the electron transport chain. Yeah, so when photosystem 2 loses an electron because a photon of light struck it, exciting that electron into its excited state, water is going to split and donate one of its electrons in order to fill the void left by photosystem 2 as that electron travels through the electron transport chain. And therefore, we have some H plus hydrogen protons and as well as oxygen, which is a really wonderful byproduct that the plants do not want, but that we need in order to survive. Okay, so those are going to be your big uses for hydrogen and oxygen. And again, oxygen and hydrogen are in all 4 of our organic molecules and macromolecules. Okay, and we'll talk about the water cycle, which is real exciting and probably something that you've talked about a bajillion times, but we'll just refresh your memory a little bit in a second. Okay, other most important nutrients, carbon. Oh my goodness. In fact, in order for something to be considered organic, it has to contain carbon and be part of a living thing. But carbon is truly the granddaddy of all atoms. And if you've had any sort of chemistry or organic chemistry, you know that carbon can form 4 bonds. It's incredibly stable. And it literally is, legitimately is the backbone of all of our macromolecules. So really, really, really important. Our major source of carbon, obviously on Earth, all organic molecules contain carbon, as I just stated, but also carbon dioxide is going to be the more common form of carbon that we'll talk about. And we can talk a little bit about cellular respiration and photosynthesis in a few minutes with the carbon cycle. What are we going to use that carbon for? Already said, the backbone of all organic molecules. Also, carbon dioxide is a reactant in photosynthesis. So we can talk about that a little bit more, but again, CO2 is being fixed through carbon fixation. And in that process, it is turned into organic material. Well, specifically, what we usually think of would be glucose, which then can be incorporated into other things. Good so far? Perfect. This is all easy. It's just a reminder for you. All right, nitrogen. So our major source of nitrogen, so we have the majority of our atmosphere, and you may or may not know this, is nitrogen, not oxygen. Oxygen's like 22%. I want to say nitrogen's like 71% of the atmosphere. The crazy thing is plants, which we rely on for everything, they're just the best. They do it all. They actually can't convert nitrogen gas from the air into a usable organic form. So they can fix, it's called fixation when they're, converting a state there and making something inorganic to organic. They can fix carbon dioxide into something usable, but they can't fix or convert nitrogen. So we're going to have to use the help of some bacteria friends for that. Major uses for nitrogen. So this is where we start differentiating because all macromolecules have carbon, hydrogen, and oxygen. Nucleic acids and proteins are the only two that have nitrogen in them. And that is something that you do need to commit to memory because I've seen AP questions in the past where they're like, oh, a nitrogen-containing macromolecule did blank, 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 blank. And whatever it did could have been like a carbohydrate or a protein, but since it's nitrogen-containing, it has to be a protein and not a carbohydrate. If that makes sense, as like a very basic example of where you may need to know that nitrogen is in nucleic acids and proteins. And that's just like some basic biochemistry stuff. Hormones also have nitrogen in them, chlorophyll, and then coenzymes, which are just going to be little chemical molecules that assist enzymes in their functioning. So all of those things are going to be important needs for nitrogen. And we'll talk about the nitrogen cycle again with the help of those bacteria that can fix the nitrogen from the air. Lastly, our last main one is going to be phosphorus. So phosphate is really just like decomposed and then taken up by plants and then decomposed and taken up by plants. So that's going to be our major source, our dead and decaying things. The uses here, so nucleic acids also have phosphorus in them. So nucleic acids have carbon, hydrogen, oxygen, nitrogen, and phosphorus in them. They have the most unique atoms in them. Obviously also ATP in phosphorus. Sorry. Oh, yeah. Phosphorus is an ATP. ATP is adenosine triphosphate, so it has 3 phosphate groups. So we need a good source of phosphorus in order to make ATP, which is obviously what keeps us alive and gives us energy every day. And then phospholipids. So remember that phospholipids are a crucial part of the lipid bilayer, which is the outer plasma membrane of all of our cells. So we need a lot of phosphorus there because they have a phosphate group and 2 fatty acid tails. So obviously we need the phosphorus source in order to construct those as well so that we can make our lipid bilayer. So lots of important reasons. And again, this is like the most important part of us talking about nutrient cycles is you reviewing in your brain the biochemistry of these major things because the cycles themselves are relatively simple and also less important on the AP exam than just knowing, again, the importance behind these atoms. Okay, cool. Here we go then. We're going to zoom through these cycles because I'm basically assuming that you guys are relatively familiar with these, especially the water cycle. I mean, we've probably talked about that forever. But if I'm going too fast at any time, just ask me to slow down or clarify. I have no problem with doing so. Okay, so water cycle. Obviously, we have lots of forms of water on Earth. We have water vapor, right? Humidity in the air. The air has a good chunk of it made up of water vapor. We have lots of sources of liquid water, lakes, oceans, rivers, all sorts of things like that. Crazy fact, 97% of water on Earth is in the oceans. So if you think of like major lakes, streams, ponds, I live right next to Lake Michigan in Chicago, so that's like major. It looks like the ocean. There's no way you can see the other side. All of that water, all the water we drink, all of that, every source of fresh water makes up 3% of water on Earth. So that just tells you how deep and terrifying the oceans really are. Anyway, I digress. So that's going to be obviously major sources there. And it just basically the water cycle is just water transforming from different states. But there are a few words you need to be familiar with. So precipitation is a big one. Obviously, snow, sleet, hail, rain, all of those things are precipitation. And that's bringing liquid water from the air into liquid water on the ground. Okay, very, very simple. But some other ones that you might be less familiar with. Evaporation, I'm sure you guys know about. Obviously, if it rains, puddles don't last forever. So the major thing there is that we're turning from a liquid state to a gas state, turning into water vapor, which is then rising. It's what forms all of our clouds, but also forms like the humidity and the amount of water in the air, which is less water in the air in the wintertime, which is why we get so dry, obviously. A couple other important words: condensation. Condensation is going to be when we have temperature changes, and that's going to be, again, a phase change, but this time from a gas to a liquid. So from water vapor to actual liquid droplets. So if you've had like a cold soda or a glass of water or something like that in the summertime, the cold air of your glass or can is actually doing a phase change, right? So the water vapor in the air is condensing, right? Turning from a gas to a liquid onto the outside of that can. So it's not water coming from inside the bottle or the can, it's just the water vapor that was in the air condensing there. And then the last one would be transpiration. So you've probably done the transpiration lab, if I had to guess. Apparently my dog wants to say hi. Say hi. Hi. No, she doesn't. But transpiration, again, you probably did the lab, and that's going to be the loss of water of plants through their stomata. So remember that there are guard cells around the stomata of plants, and they can open and close depending on the water levels. When they're open, due to the concentration gradient of water, there's more water inside of a plant than inside of the air. And so water is going to gradually be pulled out of that plant. And that process of water leaving, transpiration, actually contributes a lot to the water vapor that we have in the Earth's atmosphere. But also, even more importantly, it has to do with when that water vapor is being pulled out, again, through that negative concentration gradient, water potential. It's pulling other water molecules up with it through the processes of cohesion and adhesion, where those hydrogen bonds are sticking water molecules together and pulling them up, which is allowing nutrients and other things to move from the root of the plant up to the leaves. So it does a lot of cool things, and that's transpiration. So those are the really big Asian words that you need to be familiar with. So we have precipitation, liquid from air to ground; evaporation, liquid into water vapor; condensation, water vapor into liquid; and then transpiration, which is going to be liquid water in a plant going into water vapor. So just some words to be familiar with. You may see something like this and need to kind of analyze, but pretty simple. You've probably learned about it a ton since middle school. Okay. Carbon cycle. Main thing that you need to know about how carbon cycles through our atmosphere is that it all involves cellular respiration and photosynthesis, right? So when all organisms, including plants, right? Remember that plants also go through cellular respiration. When all living things are going through the process of cellular respiration, they're breaking down complex carbohydrates. And the byproduct of that is carbon dioxide, which is obviously what we exhale. So I always tell my students, right, if you had anything with a carbohydrate in it for breakfast, right, you're exhaling your breakfast. The carbon that was in that molecule of glucose that you consumed is actually coming back out of you. And they've tested that by having people eat like radioactive carbon-laced foods and then detecting the carbon dioxide that's coming out of their mouth, and it is radioactive. So it's able to break down and then be incorporated into a new carbon molecule and then exhaled back into the atmosphere. So then that carbon form, which is an inorganic carbon form that's, again, toxic to us, gets picked up by plants and phytoplankton and any other photosynthetic organism, and it gets reincorporated. So it goes through carbon fixation. You may remember the enzyme rubisco. The enzyme rubisco is the enzyme in plants that's responsible for carbon fixation and incorporating that carbon into organic material, okay? And then that organic material is eaten, right, by things. And then the cycle continues, right? So that's kind of what's going on there. But those are the two major processes. Something to note, and we can certainly talk about the human impact next week. I'd be more than happy to talk about it forever. But we are contributing way too much CO2 via mainly manufacturing and pollution and all sorts of things. We're burning so many fossil fuels to run cars and factories, to make technology, everything that we're doing. And so because we're doing all of those things, we are contributing a lot more CO2 than can possibly be processed back by plants, which is why we have global warming and climate change and things like that, because we have all of this extra CO2 in the atmosphere that's interrupting the carbon cycle and really leading to a lot of major problems that are needing a solution. So that's just like a fact. I want definitely to bring that to your attention, even if you're already relatively familiar with it, just because the AP exam really likes to focus on current events. I know I've said this many weeks if you've been tuning in many weeks, but that's a big one with like greenhouse gases. I would not be surprised if that shows up as a current event kind of free response this year. And you can talk about kind of the contribution to the carbon cycle, like, and how it's overloading the carbon cycle in order to get good points on that question. Okay, but again, your teachers and myself and all that, we never know what's going to be on the exam until you take it, and then we see the free response like 2 days later. So we can best prepare you, but every year they seem to focus on one topic more than the rest, and your guess is as good as mine as to what that can be. We can make educated guesses, but that's pretty much it. Okay. Back to the task at hand. All right, so we'll just zoom through nitrogen and phosphorus cycles because honestly, they are like less touched upon, but still very important. So kind of already mentioned the most important part of the nitrogen cycle, and that's that we're relying on bacteria to go through the process of fixation. So again, fixation is taking this nitrogen gas, this inorganic compound, and incorporating it, making it into an organic compound eventually. So we have fixation of nitrogen by bacteria, and it looks a couple different ways. So for the most part, we're fixing bacteria into ammonia, and we can also take a couple other forms, but mainly we're fixing it into ammonia through either bacteria or through decomposers and things like that. And then we'll go through the process of nitrification, which you can see here is turning it into nitrite, and then finally nitrate, which is something that we can really use and incorporate into the plant. The plant can also use ammonia, but nitrate is going to be another really helpful source that is coming out of this. So ammonification is part of the process of creating ammonia, but the main ones that you should know about are fixation, again, taking that nitrogen from the air and fixing it into organic compounds like ammonia, and using the ammonia, or converting ammonia into nitrate through nitrification. And then both of those forms of nitrogen can be absorbed by the roots of the plant and can then be incorporated. Again, remember that's going to help contribute to creating nucleic acids and proteins. Okay, last cycle, phosphorus, easily the easiest, which is great, because again, as I said, it's really just phosphate being transferred from dead things. So again, the major thing here is the plant is able to take up phosphate via its roots from phosphorus that's in the soil, which is also a common ingredient. Phosphorus and nitrogen are the most common ingredients in fertilizers, so you can check your fertilizer, see that. So plants will uptake this phosphorus. Remember, they're going to use that to help make nucleic acids, ATP, phospholipid bilayers, things like that. Then other animals will consume that and get their source of phosphorus that way. And then when a plant or an animal dies, it decomposes, it's broken down, and the phosphorus returns back into the soil to be reabsorbed by the plant. Another way, and it's less important to kind of focus on, but there's also things that are dead and decomposing within the ocean, as well as being, not the ocean, but a body of water, could be the ocean. There is degradation of rocks and soil and things like that. And that can lead to phosphate being in water sources that kind of just like settles out into the bottom. And then when things like waves or currents push that back onto shore, it'll end up back into soil, and plants can reincorporate it that way too. So if there's phosphorus in the water, that can also end back up in the soil, and plants can use that. Plants and animals can use that and incorporate phosphate into the nutrient cycles that way as well. Okay, again, all relatively easy stuff. The most important thing that you can take away from this is what that phosphorus, what that nitrogen, what that carbon, and what that water is going to do for the plant or organism. And again, that's just your classic example or refresher on biochemistry. Okay, so that's all we're going to talk about nutrient cycles. We're going to talk a little bit about succession and then invasive species to kind of round out the majority of the stuff that we'll talk about with ecology. And then next week we'll talk a little bit about the human impact, but more so we'll just kind of review all of these topics, and then we're going to launch into our weeks of review. So if I have to count really quick, next week we'll finish ecology, then we'll have 1 week, 2 weeks, then we'll have spring break, 3 weeks, 4 weeks, 5 weeks. And we only have 5 weeks with you guys then before AP time. So we'll spend a good amount of time on each big idea then. And again, you coming with questions is going to be the most important thing there. Okay, cool. So ecological succession. So speaking of plants needing nutrients and things like that, commonly we can kind of take that a step further and talk about ecological succession, which is the process of the changing and replacing of species in an ecosystem over time. The way that this fits kind of nicely with nutrient cycles, one, it's another thing that you need to know about ecology, but two, in order for life to start in a lifeless place, which we're going to talk about a lot today, you need to have nitrogen and phosphorus and things like that in order to make this process happen. And it is crazy to think about life starting in a lifeless place, but totally possible. It just takes a really long time, and that's something that we're going to talk about. Again, we're going to have, our plant life come first, followed by our animal life, and then this developing process. Like I had my students draw a diagram, and they thought after 1,000 years there would be like a thriving human population, and that's just not the case if we're dealing with a case of primary succession. Okay, so ecological succession, what we're talking about next, the process of the changing and replacing of species in an ecosystem over time. There's two major types that you need to be familiar with. Type 1 is primary succession, and this is crazy, but we're really going to depend on soil here for our definitions. So this is where life forms in a lifeless area with no soil. So think bare rock. Think the example that I always give is like a brand new island, right? So islands form all the time via underwater volcano explosions. Underwater volcano explosion occurs, and we have this hardening magma, this hardening lava, and that hard lava rock is lifeless. And there's no soil there, and there never was soil there, and there never was life there. We have a brand new spot. I like this diagram because it really shows you how things happen over time. So the very first step that needs to happen, and this can take 50 years or more, is that we need to create some soil, right? Because we're not going to get any plant life if we don't have soil, because they won't have nutrients. So oftentimes the first colonizers will be things like mosses, because they actually don't need soil. They can cling to rock, and I'm sure you've seen that before. We've seen moss growing on buildings or rocks or the little grout lines between bricks. So they can grow there and then start to decompose. In that decomposition, they'll incorporate nitrogen and phosphorus and things like that as they're decomposing, and that can form our first layer of soil. Then we'll have low things like grasses that will start to grow, followed by like more shrubs, and then finally trees. The reason it's going to take trees so long is because things that have such a strong vascular system like that require a great, and also just require a lot of stuff, right? In order to make a full-fledged tree, they're going to require a lot of nitrogen and phosphorus, which just isn't going to be super, super common in the environment from an early stage if you're coming from nothing, right? So that's primary succession in a nutshell. Obviously not super common, but not super complex to understand either. Okay. Secondary succession is going to be a lot easier to happen. So this is when we have life forming in an area where a community has been cleared due to a disturbance and the soil remains. So forest fires are the perfect example of secondary succession, where all living things are wiped out, but the soil is still there. And because of that, it's really, really easy to start growing things again. Oftentimes because seeds still exist in the soil or they're nearby, right? It's not like you're isolated on an island in the middle of the ocean with nothing nearby. So there are seeds nearby, the soil is there, it's already nutrient rich, it's been nutrient rich enough to grow these large species, and so it will be very easy for them to come back. So there's kind of your example again of primary versus secondary, and you can see just how much longer it would take for trees and things like that to occur in a primary situation versus secondary, where it's very easy for that life to spring back. And it's oftentimes really important. So like forest fires are actually really beneficial for forests and things like that, because usually they start because of an excess of kindling and logs and little twigs and leaf litter and all sorts of things like that. And it actually helps clear out all of that dangerous brush. And big major trees are not going to burn down very easily. And if they do, the soil is really fertile and rich then, and new life is able to spring up. So that's, again, a pretty common and classic example of secondary succession. Okay, so nutrients necessary for succession, I already touched on this, but you need a high rate of nitrogen in order to have organic material form. So you can see the soil and nutrient solutions that were incorporated in this experiment. As we increase the amount of nitrogen in the solution or in the soil, we have an exponentially higher amount of growth of organic material. And again, that's because they're using that nitrogen. It's essential for their incorporation into amino acids and nucleic acids so that they can form all the macromolecules necessary to have life. Okay. Easy enough. All right, so we're going to talk a little bit then about species diversity, kind of a segue here so that we can wrap up all major topics that you need to understand about ecology with a nice little bow. So species diversity, speaking of all of these new species coming over and taking over areas that have not had life or have had life destroyed, the health and wellness of an ecosystem is really dependent on the diversity of the species that live there. So species diversity involves two major branches that you need to be familiar with. It's both the number and ratio of different species in a given area. So the two components that make that up then, component number one is species richness, which is just the number of different species in a given area, so it's easy enough. So if you take a look at these diagrams, community 1 and community 2, they both have species A, B, C, and D. So they have equal species richness. They each have 4 species in their area. But obviously, if you're looking at these diagrams, you can determine that one is certainly more diverse than the other. And species richness is not going to be able to allow you to differentiate those, but relative abundance will. The relative abundance gives us the capability of officially saying that community 1 is more diverse. And that's because relative abundance is equal to the proportion of the different species in a given area. So obviously we are very heavy on species A in community 2 with 80% of species being species A. But in community 1 we have an equal distribution of all 4. So while we can say that yes, they have the same species richness, community 1 has a much more diverse proportion of species in that area, and therefore it is more diverse. So those are just some common buzzwords that you should be familiar with. Again, an ecosystem is going to be more thriving and more healthy if they have a higher relative abundance and a higher species richness, which leads to that increase in species diversity. They're just going to be hardier. Again, if something were to wipe out species A, community 1 would still be thriving, and community 2 definitely would not. Okay. Logan, species diversity is the number and ratio of different species in an area. So it's a combination of species richness and relative abundance. Those two terms make up the definition of species diversity. So we want to know how many species live there, and then we want to know how many of those individuals actually live in that area, right? Like, do you have just one woolly mammoth, or do you have 50 woolly mammoths? That's going to help us to determine the actual diversity of an environment. Okay. Easy enough. So what can disrupt this then, especially in communities like community 2, where they lack a lot of diversity, they're going to be much more susceptible to what we call invasive species, which are actually pretty common. And the definition there, it's pretty broad. It's any kind of organism that is not native to an ecosystem and causes harm because of this. So these species don't have to be innately harmful. They're only harmful because they're not native to that ecosystem. And I can tell you about a couple different ways that they can harm the ecosystem. But usually it's because they're bringing disease or they're out competing. So you see in the upper right-hand corner there a bunch of fish. Those are carp. And carp are an invasive species that have taken over a lot of fresh water, and they will eat absolutely anything. So the delicate food webs that are constructed within ecosystems are completely disrupted and damaged by invasive species. So since carp will eat anything, they're taking food from all sorts of other creatures and organisms, and those organisms are then dying due to lack of food. And we're losing the diversity in that area because organisms are dying out and these invasive species are taking over. You can see some zebra mussels in the bottom right-hand corner that are kind of infecting other clams and mollusks and things like that and just taking over, and they're breeding like crazy. Stink bugs in the upper left-hand corner, you may have experienced those. They're able to reproduce quickly. So oftentimes, as I said, species are invasive because they either reproduce too quickly, eat too much, or bring disease with them. And then in the middle there, which is like, I almost can't look at it because I hate snakes so much. That's a Burmese python. So that's going to be one of the longest and heaviest snakes in the world. They can average up to 16 feet and 200 pounds, which is terrifying. And this is a actually crazy invasive species in Florida right now. So they believe it started 10 or 15 years ago, probably with someone's pet getting loose, maybe during a hurricane or something like that. Obviously, it was more than one person, so massive hurricane or pet store or whatever happens, snakes escape. Ooh, terrifying. This is what keeps me up at night. But these snakes have escaped, and if you know anything about the landscape of Florida, you know that the Everglades are this protected marsh that goes on for however many acres, a bajillion. I'm going to look it up really quick because I'm so curious. How many acres? Oh, 1.5 million acres. So absolutely massive, right? And it's like this marshy area that's perfect for snake habitats. And now they're estimating there could be up to 300,000 Burmese pythons in the Everglades in Florida, which is just so terrifying. But the thing is, they're eating everything. They're eating their way through the Everglades. They eat birds that are wading. They'll eat the occasional crocodile, which is absolutely nuts, but you can look it up and it's crazy. They can take down these massive species and they're really disrupting the very fine balance of the ecosystem there because of it. Also, just relatively recently, they did a DNA test of a Burmese that they found, and they found that it was actually mainly Burmese DNA, but also mixed with another breed of snake, which was terrifying because that means they could be evolving and adapting to be better suited for the environment, and we don't need any more of them. But since the Everglades are so massive and there's just an infinite supply of food for them, they've really become a big problem there. So it's important to know about how the invasive species would tie into an ecosystem and how it can disrupt food webs and make some important connections there, because that could be definitely a free response that you could see coming, something like that, and how it has effects. Okay. So those are all of the major things that I needed to cover today. Again, we'll talk a little bit more about human impact and review all of this ecology stuff and kind of start reviewing strategies for the AP test next week. So we're going to really kind of hit the ground running there. So definitely tune in. But does anyone have any questions right now? Because we definitely have time. If you have them, I'd be more than happy to answer them. It doesn't have to be about ecology. It can be about anything. No one has any questions? Here's a question. Zachary, how advanced will the questions be on the AP exam regarding ecology? So if you tuned in last week, we did a free response from 2017, which I can post really quickly. Let me just pull it up. Biology 2017, FAQ. It's question 4. You can scroll down to question 4 and you can see kind of what that question looked like. That's relatively common. I think the ecology questions are really easy. We'll go through a lot of practice multiple choice questions from old AP practice tests next week. So definitely stay tuned. But honestly, if you understand like logic and how things are interconnected, I think ecology questions are the easiest thing that you can see on the AP exam. So if you can read a food web and see how those species would be interconnected, if you can determine a food chain, order based on a table, which is what we did last week. So you can go back and watch that. If you can determine like a predator-prey relationship or a commensalist relationship or mutualist relationship, you really should be in good shape. They're in my mind, the very, very baseline questions, usually not super in depth, which is weird because everything else that is on that exam is really in depth. But tune in next week because we're going to go through, I'll pull up a bunch, I'll make sure that I pull a bunch of multiple choice questions so that you can see those. And you can also look at last week's episode to see the ecology free response that I was talking about. I just did not, did I just post that in the group? I'm losing my mind. Oh yeah, I did. But you can also Google like AP Biology, ecology free response and see what comes up and take a look at those questions and their answers. But I think they're pretty easy, so you should be fine. Okay, what are the elements of the macromolecules? Anusha, great question. I'm actually going to type it into the chat box because I feel like that will be easier, but I'll also say it. So carbohydrates have carbon, hydrogen, and oxygen. Lipids have carbon, hydrogen, and oxygen. Nucleic acids, remember that's DNA and RNA, have carbon, hydrogen, oxygen, nitrogen, and phosphorus. So this is an easy one to remember if you're familiar with the structure of DNA. They have nitrogenous bases, right? That's what we call adenine, thymine, guanine, uracil, cytosine, right? So nitrogenous bases have nitrogen in them. And then they have the sugar phosphate backbone that keeps them connected, right? On the outsides, the really strong bonds. So those obviously have phosphorus in them. So that's an easy way to remember that that nucleic acids also have nitrogen and phosphorus if you're just familiar with the fact that nucleic acids have nitrogenous bases and a sugar phosphate backbone. And then proteins, which is the same as amino acids, have carbon, hydrogen, oxygen, and nitrogen. Because amine, an amino group, is an NH2 group that fits into the one of the 4 components of an amino acid. So those are there for you. Hopefully that helps. Anyone else have any questions that I can answer? Ooh, Zachary, this is a great question. And actually in like a screencast from long ago, we talked about this a lot. So some major differences that you need to know about, was it just DNA replication or was it transcription and translation also? Because there's more differences between transcription and translation than DNA replication, but there's still some differences in DNA replication. Yeah, I can go into transcription and translation for sure. Again, I'm probably not, okay, also transcription and translation, perfect. So I'll talk about that in a little bit and I can, or a little bit. And also talk about transcription and translation. It's not going to be as beneficial without slides, so tune back into review as well, but I'll do the best I can to verbally explain. So your major differences for prokaryotic and eukaryotic transcription and translation. So prokaryotic DNA is just a single circular piece of DNA. It is double-stranded, but it's single circular. And then we have chromosomes. So the first difference is that we have to unravel our DNA. It's wrapped around these proteins called histones. Was probably a little more than you need to know about, but we'll talk about it a little bit. So it's wrapped around like a spool of thread. And so we need to unwrap that before we can even replicate. Okay, but both prokaryotes and eukaryotes are going to have multiple origins of replication where we start this process, where we open up the DNA. And that is something that I'll probably save until I have a PowerPoint, because then I can talk to you about like 3' versus 5' ends, which is probably something that was definitely on your test today. So as far as major differences with transcription and translation, so because prokaryotes lack a nucleus, they're going to go through the process of transcription and translation simultaneously. So as DNA is being transcribed, and remember transcription is the process of going in from DNA to mRNA, messenger RNA, that as it's being transcribed, it's immediately being translated, which is the process of going from RNA to protein, because those things are happening in the same location, right? So it's like RNA polymerase is coming through and making the mRNA, and then the ribosomes are binding right away, and tRNA molecules are binding, and we're getting our amino acid chain. What that means is that also there's no RNA processing for prokaryotes. So I can explain that better probably by talking about how eukaryotes do process their RNA. So because eukaryotes have a nucleus, basically what happens is the first step of RNA polymerase coming through and making that mRNA transcript makes something that we actually consider pre-mRNA. So it's not the final message that's going to leave the cell. And so what happens is we have the spliceosome come in and remove the introns, which are the regions that are going to be non-coding that time around. Those can change. They're not always introns or exons, right? But the introns are going to be removed via the spliceosome. And then that mRNA, which is now processed and shorter, which is an important thing to know about. I've seen AP questions like that before, where they ask you how the mRNA transcript would differ if it was in a prokaryote versus a eukaryote. Eukaryotes, since we cut out regions, it's going to be shorter than the prokaryotic mRNA, which just gets transcribed holistically. So that's going to be a major difference there. Then it will move out of the nucleus. It also gets a 5' cap at the 5' end. It's a methyl group that's going to lead it out of the nucleus into the ribosome. And then it has a 3' poly A tail, which is just a bunch of adenines at the end to protect it from enzymes in the cell called exonucleases. Yeah, so that is what that region is for, and that doesn't occur in prokaryotic transcription. So the mRNA processing, the 5' cap, the poly A tail, the locations of those things are different. The length of the mRNA is going to be shorter in eukaryotic replication. All of those things would be important differences. Hopefully that covers enough, but, oh, good, Zachary, I'm so glad. I wish I had talked about this last week so that you could have had it on your test, but at least you have it for the AP exam now. And feel free to keep bringing in questions like this because it makes a lot more sense. So hopefully that makes more sense now. Okay, so Anika, hopefully that kind of briefly explain transcription and translation if you're not super familiar. But transcription, again, is going to be the process. So I always explain this to my kids. If you're transcribing a message, what it means to transcribe is just to copy, right? So there used to be scribes back in the day who would legitimately copy every page of the Bible. And I just can't picture a worse job. But we didn't have copy machines. We didn't have printing machines at that point. So they would hand copy it. That's a scribe. That's transcription. So transcription is just making a copy. It's making a copy of DNA because DNA is not able to leave the nucleus, right? It does not leave. So we're making a cheap single-stranded copy of the DNA that can leave the nucleus to then go be converted into protein. So it's basically like your messenger. So with that, what else can I kind of add? Oh, DNA and RNA are made of the same macromolecules. They're both nucleic acids. They're not the same. RNA is single-stranded and has uracil instead of thymine, right? But they're the same language, right? Just like when you're transcribing a piece of, a note from one version to another, right? You're using the same language there. You're using, the same letters and all that good stuff, the same words. So we're going from nucleic acid to nucleic acid when we're transcribing. And that's going to use RNA polymerase. So polymerase, it's an enzyme because it ends in ase. It makes the polymer. Polymer is a grouping of monomers. Think of like a tower of Legos as opposed to a single Lego. And it makes RNA. So it's an enzyme that makes the polymer RNA. So once we get the RNA, then we go through the process of translation. So if you think about what the word translation means, if you're translating something from English to Spanish, now we're switching languages. It's still the same message, or it should be if you're a good translator, right? So it's still the same message that's being transferred, right? But it's a different language that we're using to convey that same message. So in translation, now we're switching our language. We're going from the language of nucleic acids to the language of amino acids and proteins. And so the RNA will bind to the ribosome. If it's in a eukaryote, it's usually in the rough endoplasmic reticulum. If it's in a prokaryote, it's just in a free-floating ribosome. And it's going to bind to that. And then tRNA, transfer RNA molecules, are going to read the mRNA in triplet codes called codons. And they're going to match up with them and pair the corresponding amino acid that mRNA message coded for. And again, so we're switching languages. The message is still the same. The amino acids are still conveying the same message that the mRNA and the DNA conveyed, but we're using a different language. We're using a different macromolecule entirely now in order to convey that message. And the proteins are actually the things in our cells that do stuff. DNA isn't capable of doing anything, right? It just sits in its nucleus. Proteins are what actually do things, but they need to be coded for using the DNA. So hopefully that helps. Kind of hard to just like verbally explain that. I can definitely show you more pictures though. It seems like this kind of atmosphere of like genetics and genes and DNA to protein is an area that I can definitely focus on during a review. Okay, Zachary, this is such a great question. And honestly, it's a really important one. Lucky for you, there was a huge cladogram phylogenetic tree question on the AP exam last year. It was one of the long 10-point free response questions. I'll actually post it for you. AP Biology free response 2018, so that you can take a look through it. It's going to be near impossible for me to explain to you how to construct a cladogram without having diagrams, but that's something that I will make a primary focus. So our first review will be evolution. Yeah, exactly, Zachary. So the reason why I say it's your lucky day is because since it just showed up last year, chances of it showing up this year are pretty slim in such a large format. It very well may still be in some multiple choice questions. I would not be surprised at all. There's always phylogenetic trees, right? But it's probably not going to be a really hefty high points question in your free response because it was the first question in a 10-point worthy free response this past year. So you can always look at that and kind of detect some trends. So again, hopefully you don't have one this year. I did post both the question and the answer key to last year's free response so that you could take a look at that first question. Hopefully you'll find that it's actually not as hard as you think. They didn't have to construct a phylogenetic tree. They just had to analyze one. Oh, no, I'm sorry. They did have to construct a phylogenetic tree. I forgot. But it was only a few points. So, you know, on these long free responses, the averages are usually like 3 points. So as long as you can get 3 points somewhere else, you're good to go. Okay, so don't stress too much about it. If you can't construct a cladogram, but you can answer questions about a cladogram, you can still answer, 6 other points of question 1, and you're in good shape to get a passing score there. But I will make sure, Zach, that I make that a priority to cover in review because I'm not going to do it too much justice if I'm just talking about it. Cool. Love these questions, y'all. I really appreciate it. Ooh, I just made my face big. I guess I should have had my face big this whole time. It's kind of terrifying. Any other questions? I doubt that that's true, but I appreciate the sentiment, Zachary, and keep coming back for reviews because we'll talk about all this stuff, and I can answer. I mean that was quick. I answered hopefully a good amount of questions in not that long of a time frame, and this is what review will be. You can just throw questions at me, and I'll try to pull up resources real quick and get you guys the answers that you deserve. Cool. All right, y'all. On that note, I'm excited for a review over the next 6 weeks. We have lots to learn, lots to cover, and I'm very excited. So please keep coming back, keep asking questions. If you're not a 500 Plus member, $25 definitely worthy, so that you can go back and look through all of these hour-long lectures on other topics. But otherwise, I look forward to seeing you next week. And again, come with a list of questions. I'm ready to answer them. Okay, so have a great week. Happy first day of spring. Doesn't really feel like it, but it's happening. And I look forward to hearing from you guys again next week. Bye, y'all.